Welcome back. You probably recognize this illustration here as that of a mitochondrion. They're commonly called the powerhouses of cells because they generate most of the ATP that cells use. And what we're going to do in this video is we're going to take a look at the structures, so we find out about the various parts of them and what the parts do and how they're specialized to do what they do. We'll look at a theory which explains or attempts to explain where mitochondria came from, and then we'll look more closely at their functions. There's 4 commonly known or described functions of mitochondria. One of them is generating ATP, but there's 3 others, and we'll take a close look at those. And then we'll end this video with a short consideration of what happens if they quit functioning. And we have things like mitochondrial diseases. So let's get going on this thing. Mitochondria, we can use this illustration here to start. Mitochondria have 5 basic parts. They have an outer membrane, as well as an inner membrane. And right away you can see that those 2 membranes generate 2 spaces inside. We've got this space here, I'll just put a dot there and out here, so we have a space which is in between the 2 membranes, an intermembranous space. Typically it's quite thin, it's quite narrow, so the outer membrane is really quite close to the inner membrane all the way around the edges, and where the inner membrane folds and comes against itself inside, the intermembranous space there is quite a narrow strip as well. And the other space is inside here, in the middle of the inner membrane. It's called the matrix. Now I said there were 5 parts, and those are all the things I've drawn, except we can identify these layers of membranes, if you like, and they're called cristae. So this and this, these 2 pieces, cristae, one of them is a crista without the E on the end. So cristae are the shelf-like membranes inside. And I suppose they may get their name, if you think about it, if you take a look at this thing and narrow out the spaces a little bit and draw it vertically, you wind up with an appearance that may look more like that. So it looks more like a bookshelf than anything else. So these cristae then are the membranous structures inside. So those are the 5 structures I said there are, but there's actually a couple more things we can identify in these mitochondria. One of them is the fact that they have their own DNA, mitochondrial DNA. And the mitochondrial DNA, and I'll be very careful how I draw it here, is actually circular. DNA we typically draw like this, as a double helical strand with the bases attached to each other by hydrogen bond. But in this mitochondria, they're actually circular DNA, which is reminiscent of bacteria, because bacteria have circular DNA. There's also other DNA strips that's more like this, like that one. But this one is an important one. We'll get to that in just a second. And the other thing that we can put in here is, I'll use green here, the fact that they do have ribosomes. So this is mitochondrial mtDNA. Mitochondrial DNA. When you take a look at that, they have their own genetics then, if you think about it that way. So they have their own genetics, their own genome, and they can, because of the presence of the ribosomes, they can actually make their own proteins, which is rather unique, very interesting about an organelle that can actually make its own proteins. They can even divide, they can replicate themselves within another cell. And that kind of gets us to the topic of where they came from. And it's really quite theoretical. And we'll take a look at that, and it zeroes in on this relationship between this circular DNA and bacteria. And just before we do that, though, we want to mention, I want to mention that you can't really see these in a cell. When you look at a cell under a microscope, what you see is the nucleus, and sometimes you see a different staining part in the nucleus called a nucleolus, and so you can see bits and pieces of things in here, but you can't really see mitochondria because they're quite invisible. They're big enough to see, so you should be able to see them in a cell if you're really magnifying it, but unless they're stained, they don't show up. So you can't really see them. And how many are in there depends on the cell. If we're talking about a blood cell, like a red blood cell, they don't have any. Red blood cells do not have any mitochondria. They do not generate their own energy. They wear out, they die. A liver cell, a hepatocyte in contrast, can have thousands of mitochondria. Thousands of these things inside a single cell. Liver cells, hepatocytes as they're known, are very, very active cells. They need a lot of ATP. So they have a lot of mitochondria. That allows us to understand how they can make copies of themselves and replicate. The other thing we should mention before we look at the theory part is that this is what we typically allow them to look like if we sketch them. They don't always look like that. They are represented as elongate, sort of sausage-shaped organelles, but they can actually be very long. They can actually join together with other mitochondria. They can divide, they can separate from other ones, and they can actually change shape. So this picture is quite generalized, and they obviously specialize and do slightly different things as the cell's needs may change. So that's just general information about what mitochondria are like. How do they get there? That's the theory. Let's consider this as a cell then. Just an ordinary cell. I don't care what cell. And here's a bacterial cell. I'll just erase that. Bacterium. What we had in a previous video, and you'll know this anyway, is that these cells can actually engulf that. So we have a process of endocytosis. So what can happen over a short period of time is this cell here may get an invagination, as it's called, and if that's the first picture here, and that's the second picture, and the third picture over here, I'm going to color this dark, so the bacterium is dark. This is the bacterium here, and coloring it dark here. That's what they believe happened. This thing becomes known as a mitochondrion. They believe that many people believe that bacteria were taken in eons and eons ago by cells, and they formed a symbiotic relationship. So the bacterium, which does produce ATP because bacteria have to survive, the bacterium now is specialized inside a cell. It produces the ATP, which the cell can use. And over the eons of time, when this may have occurred, historically speaking, is this bacterium here has, its outer membrane has become very convoluted, and it has done this. So if we take a look, and I'm going to erase everything that wouldn't be bacterium. This membrane here is really part of the cell membrane of the cell. And what we're left with, if we erase that, we're left with something that sort of resembles a bacterium. And sure, it's not the right shape because the outer membrane has become very, very convoluted, but it does have the kind of DNA, because bacteria have bacterial DNA. It does have ribosomes, because bacteria can generate their own proteins. It does have a matrix, matrix is this, which is really the bacterial cytoplasm, or is what believed was the bacterial cytoplasm. So that's what sort of the theory is. This is called the endosymbionic theory. Endo meaning inside, and symbiosis. Symbiosis is that long-term relationship between two organisms where there's a mutual benefit. Mutual benefit here, the bacterium doesn't have to want for anything because the cell will supply it, because the cell always has chemicals in it and it can supply what is needed by the mitochondria now, and the mitochondria produces the ATP for the cell. It's only theory. It may be true. It seems quite likely. And one of the other pieces of evidence that allows people to believe that it might be true is this membrane here, which is the bacterial membrane, or the inner mitochondrial membrane, and this membrane here, which is the outer mitochondrial membrane, is really part of the cell membrane. It was generated because of this invagination here. So we have two different membranes, and they are really quite different, structurally different, because this one, I'll just erase this and illustrate it here. If we take a look at this as the surface of a mitochondrion now, and this may be the part of the inner membrane. So this is the outer membrane, and this is part of the inner membrane. These two membranes are very, very different. They are structurally different, distinct in some very, very obvious ways, because this one has certain proteins called porins. A porin is a protein that lines the nuclear pores around a nucleus in a cell. So if they have the nucleus here, we know that the nucleus gets protein into it because it has to do things. Enzymes go in for DNA replication, things like that. So this porin will allow proteins into the space here. So the proteins can get in here because the mitochondria needs proteins. These porins don't exist on the inner membrane. There's no porins. So the outer membrane has porins for transporting proteins, no porins. But it does have transport proteins, other transport proteins. So there's a distinct difference between their functions as well. And we'll get into exactly what that's all about when we deal with how they're making the ATP, because that relationship is important for that. A couple other things that we should mention here before we have a look at the actual functions of these things is that this DNA that's inside here, the mitochondrial DNA, if we go back to not the endosymbionic theory, but we go back to reproduction as a topic, and we talk about human reproduction. We have an ovum, and we have a sperm cell. Not a very good sperm cell, but you get the idea. The DNA is up here in the head, mitochondria in the mid piece here. When fertilization occurs, the DNA enters the chromosomes enter the ovum. That's all that goes in. So this is just DNA that goes in here. Just the chromosomes. 23 chromosomes. All of the organelles that are in the ovum, and after that in the fertilized egg, the zygote, all of the organelles are maternal. They all come from the mother. They all come from the ovum. So the DNA that's inside a mitochondrion, the mitochondrial DNA, is actually maternal in origin. It didn't come from the father, because the only thing that entered from the father is the chromosomes. It's maternal. And when you get into ancestral tracing, DNA tracing, that sort of thing, that's the DNA which they sometimes look at in terms of determining ancestral relationships, is the mitochondrial DNA, because they can trace it from mother to mother to mother that way. Okay, now it's time to have a look at the functions. I said at the outset there were 4 main functions for mitochondria. The first one we already mentioned is generating ATP. And I think I can use that picture. I don't need this here. I think I can use this picture here. I'll just finish it a little bit. In general, we take the outer membrane. It is not convoluted. The inner membrane is convoluted. I haven't said anything about why the inner membrane is convoluted, why it does all that, and it's simply because of surface area. If it was just a single membrane inside, it would only have a certain amount of surface area, but by being bent and folded all around inside a cell like that, the surface area that's in here is tremendous. If you were to take that and try to stretch it all out, it would go a lot further than just across that cell, obviously. By having more surface area, these mitochondria can have more sites for enzymatic reactions, because the enzymatic reactions and some of the processes involved in making the ATP occur on the surfaces of these membranes. So we have something like that. I'm not trying to represent ribosomes here on endoplasmic reticulum, but I'm representing proteins inside that membrane. So I'll just give you a name, like this protein could be called a cytochrome. That's the name of one of the, the name of one of those kinds of proteins is a cytochrome. So now what happens to generate ATP, remember this is the mitochondria, out here is cytoplasm. Cytoplasm, fluid of a cell, gets sugar in it, we have glucose, and glucose begins this carbohydrate metabolism process, and that's a topic in a totally different video, but we wind up with acetate ions, which are 2 carbon ions, and because of the activity of coenzyme A, they get transported into the mitochondria. They'll get through the outer membrane, they get through the inner membrane, they get into the matrix. So this 2C, the acetate ion, I should mention, we have that much, if we think about it, two carbons, we could have the equivalent of 6 hydrogens associated with it, but with acetate it's not quite, because there's oxygen involved. But you get the idea. You get this 2 carbon thing in here, and what happens inside here is the activity of Krebs cycle. Krebs cycle is where this 2 carbon acetate bonds onto a 4 carbon molecule called oxaloacetic acid, which is inside the matrix, and we get the 2 carbon, I'll put it this way, 2 carbons plus oxaloacetic acid, which is a 4 carbon molecule. We wind up with a 6 carbon molecule, which is known as citric acid, and then citric acid loses the carbon, loses the carbon, loses the carbon, so we wind up with a 5 carbon, back to a 4 carbon, and back to oxaloacetic acid. So that's basically what Krebs cycle is like. And every time it loses a carbon, it can generate carbon dioxide. And that's one of the reaction products of cellular respiration, because cellular respiration takes the glucose with oxygen present, makes water and carbon dioxide. We haven't said anything about the water yet. Where's the water come from? Well, I drew this here so you can recognize there's a lot of hydrogen involved in this. And I'll put the hydrogen there, although it's not quite like that. What happens is when the carbon dioxide is generated, the hydrogen, which is freed up from Krebs cycle in here, gets pumped, gets literally not pumped, but moved across these cytochromes. So hydrogen ions wind up into being put into this intramembranous space. What that does, obviously, is it creates a ionic potential. You put charged ions out here, there's a potential difference then. almost like an electrical difference, but in almost a pH difference. So that generates, there's a concentration difference. And with that concentration difference, as the hydrogen continues to build up out here, there's a greater potential for something else to happen. And that something else is making the ATP. The only thing is, as these hydrogens get moved across here, they become energized. They are moved, and they get moved back in. And I'll put a blob down here. This is a particular enzyme, and it's called ATP synthase. And what happens, quite simply, is that these hydrogen ions, which are generated inside the matrix and pumped into this space here, which is the intermembranous space, they get moved back in here. And as they move back in here, they lose their energy. This captures the energy off them. Put a plus here. Captures the energy from the hydrogen ions. And what does it do with the energy? It has to put it someplace. And the energy then obviously is used to make ATP. Because energy is required to bond that extra phosphate group onto adenosine diphosphate. Remember, this is adenosine, which is the adenine base, ribose sugar, and 2 phosphates. To put that additional phosphate on takes energy, and remember that the phosphate-phosphate bonds are high energy bonds. So there's the energy, it's locked up right there. So these things make the energy in cells. How do they do it? Well, it starts out here, obviously, gets into Krebs cycle. Krebs cycle degrades the carbon molecule, generates carbon dioxide, which is a waste product, which eventually gets exhaled. The hydrogens are left behind. The hydrogens become energy-rich ions, and the energy is captured by this enzyme, which allows that energy to be used to generate the ATP. So now I've erased my big mitochondria, and I've replaced it with the chemical equation for cellular respiration. I just want to use this to summarize this ATP production by mitochondria. We start with glucose, this is in the cytoplasm, and we have glycolysis, but this reaction is a summary. So what really goes on is C6H12O6. There's 3 things that happen. There's glycolysis, and the product of glycolysis, as we just used the word a little bit ago here, is the acetate ions. Now, acetate ions are 2 carbon molecules, and this is a 6 carbon, two carbon ions. This is a 6 carbon molecule. So in glycolysis, we actually have some carbon dioxide that's given off. Glycolysis generates 2 acetate ions. So there's a total of 4 carbons. 2 of the carbons are given off in glycolysis. These acetate ions, as we said, go into the mitochondria, where they're treated by Krebs cycle. Krebs cycle is where the rest of the carbon dioxide is generated. So we have a total of 4 carbons, 2 times 2, 4 carbons going into these reactions of Krebs cycle in the matrix of the mitochondria. So Krebs cycle will generate the other 4 carbon dioxide. So we've got our 6 carbon dioxide that way. The other thing that happens in here, as we mentioned before, is that the hydrogen ions go into the intramembranous space, and they have the energy. The energy gets captured by ATP synthase. ATP synthase, or ATPase, I should back up a little bit here. ATPase itself is a group of enzymes that are involved in generating ATP. ATP synthase is the one that is in the mitochondrial inner membrane, and it captures the energy out of the hydrogen ions. And in doing so, it makes the ATP. So we have ADP plus the phosphate, and that makes the ATP. And that's all fine and dandy, but what I haven't said anything about yet is what happens to the hydrogen ions. The hydrogen ions get moved from the matrix into the intermembranous space, and then they go through this ATP synthase enzyme back into the matrix. Well, lo and behold, inside the matrix, there's oxygen. And oxygen picks up the hydrogen. So we have 1/2 O2, really half of an oxygen molecule, plus a hydrogen ion, which becomes an atom, really. So we have two of these. We'll write it this way. It's a bit of an oversimplification, but that's where the water comes from. So oxygen is the final, what we say, it's the final hydrogen acceptor. The hydrogen's been the key in the whole thing, making all this ATP. And I'll tell you right now, there's 38 ATP molecules are made in this process. And if there isn't any oxygen available, this whole system here backs up. It shuts down, because if there's no oxygen, there's no place for these hydrogen ions to go. The hydrogen ions sort of plug up the system, and it stops right there, and we have anaerobic respiration, which we've also called lactic acid production, and it's very inefficient fermentation processes. So that's the first of the functions of mitochondria. The first one is ATP production. And it's a fairly complex process, but we'll leave it at that. The second one we want to mention is cell death, actually, and it's called apoptosis. And the fact is that cells die. They get worn out. Liver cells will get worn out. Red blood cells, they don't have mitochondria, but they wear out, they die. Their membranes simply wear out. The hemoglobin molecule will no longer transport oxygen effectively, but our skin cells will die. The skin cells on the surface are dead cells. We have muscle cells that can die. We have all sorts of cells. They simply wear out, and when they're not functional anymore, they die, and that process is apoptosis. The way this works really is it's a function of the mitochondria. The mitochondria, if you remember, they have these cytochromes. The cytochromes are those molecules on the inner mitochondrial membrane which the hydrogens go across as they're moving into the intermembranous space from the matrix. What happens when the cells wear out is the mitochondria release the cytochromes. And when they release the cytochromes, that leads to the cell death. Third function is calcium storage. Mitochondria will accumulate and store calcium ions. Now, calcium is really, really important in the body. It's used for blood clotting reactions. It's used in nerve impulse transmission. It's used in muscle contraction. It's used for building strong bones and teeth. So there's lots of cells, if you think about it, nerve cells, muscle cells, bones and teeth, so bone cells. A lot of cells in the body need calcium, and mitochondria will store it, and as the cells needs manifest themselves, they can release calcium, and cells can use it that way. And a fourth function is to generate heat in cells. Thermogenesis, generating heat in cells. You think about all these reactions that happen here. There's a great long set of metabolic pathways, and although enzymatic reactions are very efficient, there is a little bit of energy loss at some time, at some points along the way, and that is used by cells wisely because it's actually heat. So we have a thermogenetic component to mitochondrial function. So that sort of wraps it all up, except for the very, very last topic. I said at the beginning I want to mention something about what happens if they quit working. I mean, there's a lot of things that happen because of mitochondria, and if the mitochondria don't work properly, then some of this stuff won't happen. And I guess you have to think about the fact that there's mitochondria in all your cells except red blood cells, really, and the mitochondria in liver cells give the liver, for example, I'm going like this, the liver's down here, lungs are up here, they give the liver the energy for them to do their functions, and the muscle cells, the mitochondria in muscle cells generate the ATP to allow the muscle cells to contract and that sort of thing. And if the mitochondria aren't able to do their function because the proteins are not being generated properly, there may be a genetic defect that doesn't allow the mitochondria to be produced properly, those cells won't work properly. Mitochondria can't produce the energy, those cells won't work. So a mitochondrial defect could look like a liver disease, or a mitochondrial defect could look like a muscle disease. And the fact is, it could be the exact same defect, the same genetic mutation, the same DNA error in the mitochondrial DNA or in an enzyme that is being used in the mitochondria. The same genetic error could look totally differently depending on which tissue type the disease or the disorder is in. I think that wraps it up. Mitochondria, very interesting, very complex little organelles in eukaryotic cells. Thank you very much.